Hi everyone. Today we have Dr. Erin Greaves, who is joining us from England. She is a researcher there at the University of Warwick. She received an EndoFound grant from us in 2018. And today she's here with us online to just talk about the research she did with the funding from EndoFound, as well as her other research. So Dr. Greaves, I'm going to leave it up to you to introduce yourself.
Okay, thank you. So I'm an assistant professor at Warwick Medical School, University of Warwick in the UK. And my lab, the Greaves lab, are investigating mechanisms underpinning endometriosis pathophysiology with a specific focus on the role of immune cells called macrophages. But we also work on the refinement of preclinical models that will hopefully aid the discovery and testing of new therapeutics.
Great, thank you. And could you tell us a bit about what inspired you to conduct research on endometriosis?
Yes, sure. So I was really inspired to work on endometriosis because it's such a debilitating condition and it affects so many women. It's so widespread, yet very little is known about it. And obviously treatment options are very limited.
Okay, great. And so could you just tell us a bit about the project that EndoFound funded and where you are with that project now, what the findings were, things like that?
Yeah, sure. So as you mentioned, I received the EndoFound Award in 2018, and the funding was for a project called a single cell discovery project to compare mouse models of endometriosis and shed light on different theories for the etiology of endometriosis. So I applied for the money because I previously developed a unique preclinical mouse model of induced endometriosis that aims to mirror the process of retrograde menstruation, which is one of the leading theories for how endometriosis develops. And we want to try and produce endometriosis-like lesions in recipient mice. But mice don't normally menstruate, so we have to manipulate the animals in order to artificially induce menstruation. Now, we've used this model to explore disease mechanisms of endometriosis in the lab for quite some time now. We initially published the model in 2014, but there are many different kinds of mouse models used by the endometriosis research community, and we think that the variations could be a source of slow advancement and problems with reproducibility in the field. So during some refinements that we were performing on our model to implement non-invasive imaging of endometriosis lesions, we noticed some key differences when we compared different mouse models to each other. So when we used naive endometrium, so this is from mice that have not been manipulated at all, compared to menses-like endometrium, we found that there was differences in how long the lesions survived in the mice and also how bright they were when we imaged them. So the aim of the project funded by the EndoFound was to really explore why these key differences in lesion survival occur when different endometrium is used to produce lesions, and we used single-cell RNA sequencing to analyse the cellular composition and gene expression profiles of the different kinds of endometrium, but also the lesions that they produced.
Mm-hmm. Mm-hmm. And then as a result, then what were the findings of that study?
So the funding for the project has ended now, but we're actually still doing a lot of the analysis. Mm-hmm. But so far, what we've been able to identify is that the cellular composition of the donor endometrium and the resultant lesions differ significantly between models. So we're actually doing a lot of dry lab work now, bioinformatics processing of the sequencing data, and we are ascertaining differences in the gene expression profiles. We're currently validating these gene expression changes in fresh samples, but also comparing them to human endometriosis lesions recovered from women, such that we can determine which of the models is most representative of the pathology in women.
Okay, yeah, and that brings me to actually, why is it important to do these studies like the one that you're describing now?
Yeah, so we think that this is really important because we need to really use models that are most representative of the condition in women. We need to be looking at lesions that are the same as what we recover from women with endometriosis. And the reason that we have to do this is because we need the most physiologically relevant preclinical testing platforms that we can possibly produce. And I think we're still a long way off this, but being able to know how the models differ from each other and which one most closely recapitulates the condition is really, really important for testing therapies, but also identifying new therapeutic targets.
Yeah, yeah, exactly, definitely. And I imagine that there are some challenges that come with a project like this. So what challenges have you encountered during the process?
So actually one of the main challenges that we had generated a lot of RNA sequencing. But in my old institution, we actually had a shortage of bioinformaticians that could actually analyse the data. And actually the timing turned out to be really great because last year I was recruited to a new institution, the University of Warwick. And the computer science there is really very strong. So I had a lot more exposure to people working on bioinformatics. So we've been able to perform much more in-depth analysis and use unique methods that we didn't previously have access to. So the initial problem actually turned out to be a benefit in the end.
Yeah, that's good. Great. And I think another thing I want to ask about the project, and then I'll move on to other projects you're working on, is what do you think the impact of the study would be on the endometriosis community?
Yeah, so I think the work that we've done is going to be very beneficial to other academics working on endometriosis. So the plan is that we will make our data freely available so that other researchers can mine the data for use in their own work. And once we've finished the analysis, we really think that the work will shed light on how different mouse models can be used and which of the two investigated really matches the etiology of endometriosis in women.
Mm-hmm.
So the grant has also been massively important to the work of my group. So we've learned so much about our models and produced a really comprehensive library of the cellular composition and gene expression profiles of the different types of endometrium that we've used and the resultant lesions, which has given us really key insights. And it's a resource that we will use for many years to come. And we think that many new projects will stem from this data as well.
Yeah, that's great. I think that has the potential to have such a big impact on other researchers who are doing on endometriosis research and using mouse models for their projects. So this is very big. So I'd also like to hear about some other projects you're working on. I know that you have I know your main focus is on macrophages and their role in endometriosis. So could you tell us a bit about that, and especially why it's important to look at macrophages in the role of endometriosis?
Yeah, sure. So the majority of my research funding comes from the Medical Research Council in the UK. And as you mentioned, the main focus of our research is on the role of macrophages. And these immune cells are so intimately involved in the pathophysiology of endometriosis because they control many different components of the lesion. They're important for the establishment, they promote the growth, endometriosis lesions. They promote infiltration of blood vessels and nerve fibres, and this contributes to pain in the disorder. And we've also shown that macrophages produce factors that activate the nerve endings and do actually cause pain in endometriosis. So at the moment, what we're trying to determine is where the macrophages come from. We know that they have different origins, and the origin of macrophages actually dictates their role in disease pathophysiology. So it's important to know where they come from. And we're using this same technique, single cell RNA sequencing, to look at how many different types of macrophages there are in endometriosis lesions. And so the plan is ultimately to identify which type of macrophage is the disease-promoting macrophage and what makes that unique to all of the other macrophages in the body. So that hopefully we can target those disease-promoting macrophages using a therapy.
And could you tell us a bit about, you were talking about some of the other research you've done related to macrophages. So I know, for example, last summer in 2019 summer, you had a paper published on, again, macrophages. Could you tell us a bit about that work and why the findings were significant?
Yes, I did touch on that briefly. So in that paper, we were able to identify that macrophages in endometriosis become disease modified so that they produce high levels of certain factors, and one of those factors that we identified was IGF-1. So this is insulin like growth factor 1, and we were able to show that macrophage derived IGF-1 promotes nerve growth into lesions, and that same factor can also activate the nerve fibers and make them hypersensitive. So following exposure to IGF-1, they express increased levels of ion channels that are really important in pain perception. And we used a combination of human tissue, cell models, and a mouse model to explore this mechanism. And we were using an IGF-1 receptor inhibitor in the mouse model. We were able to show that these mice with induced endometriosis had reduced levels of pain-related behaviours.
So then, I mean, we're really seeing the big role of macrophages on pain in endometriosis, right?
Yeah.
So is there, could macrophages then be a target in the future when developing treatments for endometriosis?
Yeah, so we really think that they can be. And actually, we are recruiting for a new project that will start in September or October, looking at how we can actually target these macrophages using nanoparticles to deliver specific therapies directly to disease-modified macrophages. So this is obviously at a really early preclinical stage, but we're really hopeful that we're going to see some really significant results using these experiments.
Yeah, well, we're hopeful too. If you're hopeful, we're hopeful too. Okay, so I also just want to ask you a few other things. So particularly, like, what excites you most about your work on endometriosis?
So I think recently, now that we have the new lab up and running at my new institution, we've had some really great breakthroughs with the macrophage work, and it's so exciting when you get those breakthroughs in the lab, and it's really great for everybody. It's a really good morale boost because science doesn't always go the way you hope they will go. So getting really good results that are inching you closer to a result is really exciting. and that feeling of gradually getting closer to something that might translate to a new therapy, or to help others produce a new therapy is very exciting.
Yeah, definitely. Just like what you're saying about the macrophages now and potentially targeting those macrophages. So that is very, very exciting. And also, what research do you feel, needs to be conducted in endometriosis? Further research in your field, do you feel needs to be done?
Yeah, so I think there still needs to be a lot of work on refining and standardizing preclinical models. We know that mouse models don't truly recapitulate the disorder because we're not really able to model all of the different subtypes of lesions. So most mouse models only really model superficial peritoneal lesions, although there are some newer models that have been recently published over the last year or so that are really trying to mirror the spread of lesions, but also using techniques to make them more invasive as well, to try and recapitulate deep infiltrating lesions. But we also really need to focus on why lesions cause pain. I think the pain mechanisms of endometriosis they're still quite poorly understood, especially why women present with such varying manifestations of pain. We really need to understand what kind of lesions cause pain and how this can be better targeted.
Yeah, so we have a long way to go with endometriosis, but we're getting there, like you said.
Yeah, definitely.
Yeah, and I guess on like a closing note, the grants that you received from the Endometriosis Foundation, from our foundation, what has been the importance of this grant for your work?
Yeah, so it's been massively important for our work. And we know that a lot of new projects are going to stem from the data that we've generated. So it's been an amazing foundation to launch new projects, for getting new trainees to follow genes of interest. So it's been massively beneficial.
Great. We're really happy to hear that. And do you have any final thoughts or comments?
Yes, definitely. So I would really like to thank EndoFound for our funding. It's produced data that's going to shed new light onto mouse models of endometriosis, how they vary, and their ability to mirror the condition in women. And the data is really a key resource to other people exploring disease mechanisms of endometriosis using mice.
Great. We're so happy to hear it. Thank you for your time.
Thank you.